Thank you. The fact that we're zooming this, we're recording it, and we have a live audience is a big thing for our chapter. First time for all three of those. But I think it's a great night for that with our 6 speakers. These are students and researchers who are from the Southland here and are doing great work. And I think we're okay. I think we're going to enjoy what we're going to have tonight. Tyler's our first speaker is Tyler Zarubin, a PhD student at UC Irvine's Department of Ecology and Evolutionary Biology, working in the Kaylin Mooney lab. But he's also linked to Concordia University because that's where he works. He's assistant biology professor there, but also he has his test plots there, which you'll probably see an image of tonight. His presentation tonight attempts to enlighten us regarding a plant's water use strategy when confronted with precipitation variation, how those changes affect the insect communities that depend on those plants, or in one way or the other. So his presentation title is Plant Herbivore Interactions and the Forces that Mediate them. Tyler.
All right.
You just tell me when you want.
Okay, yeah, you can go ahead and put it on out there. We'll get started. So first, thank you to the chapter for inviting me to give this talk. I really appreciate it, Dan, especially. I'll give you a bit of the connection and I'll get started with my talk here. I'm work at Concordia University at Irvine, and as I was coming into Concordia University Irvine, Brea Dam was on her way out as retiring professor in English. And so her and I made a connection because I do manage the Heritage Garden, which we and some of you know of that. So all right, so we can.
There is a problem.
There's a problem.
Tell a joke, sing a song.
Couple other things. I just got back from Maui. It was awesome there. That's why I'm wearing my sandals and my Hawaiian shirt. I just can't let it go quite yet. But it was sunny today, so I heard it was been gloomy the past 2 weeks. Really enjoyed my time over there. I use iNaturalist a lot. And I saw some of you had the app out. I was able to find some really awesome cowrie shells and some other things in that way. So I look at Flora and fauna. I'm a graduate of UC San Diego, College of Behavior and Evolution Department down there. And then I got my master's over at Colorado State University, and now I'm a PhD student at UCI, and I work at Concordia. Looks like we're all set.
Yes, thank you.
All right. Okay, so there's my title. I'll go ahead and move on from that. Advance the slide.
You do it manually, that's not.
Yeah, go ahead with that. All right, so I'll just give you an overview of what I am going to talk about tonight. My interests are how changes in precipitation really affect plant growth, and then how those changes in plant growth mediate herbivore density. So this is really kind of the big focus of my PhD research here is how does plant performance change in response to changes in water? And then how do these changes then scale up to affect the plant herbivore interactions? As I do this, I'm going to talk a little bit about some of the methodology or some of the background actually. Advance the slide.
Just say next.
Next, please.
Again. Click on the screen.
So plant defense strategies is what I want to speak a little bit about.
Yeah, there we go.
All right, plant defense strategies. So 2 main strategies here that I want to talk to you about. Go back on direct and indirect. So direct defense strategies are those that increase plant resistance by reducing the performance of herbivores. And whereas the indirect are defenses that decrease herbivore abundance by recruiting their natural enemies. And this slide kind of shows a little bit of that. So direct defense, one example of these might be terpenes that we find in California and many other plants. We also have some indirect defense mechanisms. Here would be the volatile organic compounds the plant would secrete in order to attract parasites that then will feed on those herbivores, or complexity maybe as a means to make it easier for birds to prey on these insect herbivores. And so these are the types of defense strategies that I'm interested in that serve as a background for my study. I'm also interested in plant resource use theories. And so resource use mainly from the perspective of water. So these resource theories, they seek to predict how changes in plant resource impact the plant defense and ultimately herbivore abundance. A couple I do want to talk about with you. The plant vigor hypothesis and the plant stress hypothesis. And so these are two different theories on how plants would respond, let's say, to a decrease of water levels. So for example, if we have a plant that is facing a drought, then the plant vigor hypothesis predicts that this plant will decrease its performance. So it will lower its fitness. It's just not going to be as healthy, and as a result, it's really not going to have high effect herbivore density on it. Whereas the plant stress hypothesis kind of predicts the opposite result here. Plants, air adapted plants, they might maintain their performance by sacrificing investment in defense. And because they sacrifice that investment in defense, that might lead to actually a greater herbivore abundance and diversity. And there's evidence to support both of these. What we're really interested in here in this study is to look at how different plants native to Southern California region, the coastal sagebrush community, how they would respond to changes in precipitation, and if any one of these theories may be supported through the data that we collect. So my research questions, there's three of them really here. The first question really centers on plant performance. So how do the plants actually respond to changes in water availability? And so I'm really interested in traits that I'm labeling as functional traits. So really that's plant specific. The second question is then how does the quality of the plant change, and the quality from the perspective of the insects that eat it. So I do assays where I feed plants in my treatment plots to a generalist herbivore and just measure masking as a function of plant quality there. And the third question is the question that I'm most interested in. Advance to the next slide. And that question is plant herbivore density. And are these differences in density, is there evidence to suggest that they are mediated by the functional traits that are in response to the water that I'm giving them? And so I'll spend most of my time tonight kind of talking about that. I want to give you a little bit of background on my plot design. So I have 20 plots, and this is Vector University in Irvine. If you don't know where that is, it's right across from the street from Tanaka Farms. So I have space there that the university was kind enough to provide for me. These plots are about 6 meters by 5 meters in total. And in these plots, I planted one plant per species, 14 species total. I'll give you a species list in a moment. Grew them up in a greenhouse from seeds for a year, planted them in February 2020. I put weed barrier over the whole plot. And it turns out that weed barrier is really good at keeping moisture locked in the soil. And my studies on varying moisture. So you can see how that worked out. And I discovered that in February 2022. So I said, I think we need to remove all the weed barrier. So I did, and they've been running a year without it. And here's kind of a plot map that you can see right here. So the different shades, ambient is where I add no water, and the darker is 2 times ambient. And so what I'll do is I'll look at monthly rainfall totals, and then I'll add twice that amount, double that on top of the plot, so they're all irrigated. And this is just kind of a layout of the different plots after gardening for you. Um, All right, so here's my species list. So I'll give you a moment to take a look at these. The species that I chose, or the reason why I chose these on this list were a couple. One was availability, and two was to kind of coincide with some of Sarah Kimball's work that she's been doing on restoration in Crystal Cove area. And so you kind of look at them there, but I'll move on because I got 2 minutes left to the methods. And so I'm interested in plant functional traits and I'm not going to go into what I did with those, but this is how I actually assess the arthropod. So we use a vacuum sampling method as opposed to bead sheets or some other ways of collecting the arthropod. So I take a rigid shop vac with a portable generator, and we go out every May and we vacuum the plants. We stick a mesh bag in there, it's a mosquito net, and we just vacuum for a couple minutes and kind of suck up all the insects that way. And then what we do is we take these this plant shaft, and then we take all the insects out of that plant shaft. And you can see right here, these are all insects in a petri dish. Most of these are thrips right there, and I put them in little grids of 10. So what else to do? So we sort them out, and then what we do after we do that is we classify them down taxonomic order, or for the hemiptera, we go down to sub-order because that order is so diverse. And then the orders after this, we group them up into feeding guilds. And then we'll analyze them both from a perspective of order and from the perspective of feeding guilds. So I've been doing this 3 years now. I'm on my third year data collection, still working through the summer to get that done. I would say that we've sampled about 40,000 arthropods, classified them down to taxonomic order. And I really haven't looked at the data yet. That's something for later in the summer, but we'll see kind of fine. Maybe I'll be back here at some point and kind of present on that. So practical implications, improving our understanding of arthropod abundance and diversity in coastal sage grouse communities. We know very little about that. Compared to the plant and bird abundance diversity, even that we'll get some more information on. And then the second one here is a more comprehensive picture on how water variability changes affect the plant arthropod relationships in these communities. And I think there's restoration implications for that. A lot of restoration you choose to irrigate or not irrigate. And I think this study can help address the implications of that. So I want to thank you very much. There's my email if you guys can contact. Right, one more question. Anybody has it? Yes, sir.
Great talk. The question I had was, based on the assumptions going from your plant stress hypothesis, would that suggest that the abundance and diversity of insects in a community would reach some sort of peak at some level of moderate drought stress due to the reduction of?
Yeah, I think that, yeah, is it like an intermediate level there where maybe in that middle zone of, as I'm adding water, might there be a point where, like, in the mid zone, I have the most abundant insect. I don't really know. I mean, I think so, but I think it's going to vary based on all the plants I have up there. They're all perennials, but they're responding very differently to water. Like the Staphy's aegigoides, that really likes water, and it doesn't do very well in the ambient plots. Whereas some of the others, they don't like the water. I think some of the Areogna doesn't seem to have done very well. So that's kind of what I'm finding. But yeah, to be determined. All right.
Thank you.
Thank you.
Tyler. Thank you. Thanks, Tyler. Our second speaker is Haley Heesch. Heesch, because it rhymes with Heesch. Let's see, Haley Heesch. Let me see if I can actually read this. Use your vine. A light of sense? No.
Your phone was brown.
Yeah. I don't have it. There we go. Thank you. No problem. Yeah, before working for the California Department of Fish and Wildlife in San Diego, she received her bachelor's degree in biological sciences at UC Irvine. She's now a full-time field researcher at UC Irvine's Environmental Collaboratory Department, where she examines and evaluates the health of various vegetation communities throughout the Orange County area. She also helps run the UCI Center for Environmental Biology's undergrad intern program. So that's why she knows a lot of the UCI folks, students and so forth. Her presentation tonight is based on a thoughtful response to the 2020 26-acre fire that swept through the Bolsa Chica Mesa, exploring which restoration techniques might be most effective in post-fire restoration efforts. So the title of her talk, Post-Fire Restoration at the Bolsa Chica Ecological Reserve. Haley.
Can everyone hear me okay? Thank you for the intro.
Getting there, don't fight me.
Oh, no worries.
I'm using the pointer, so. There we go.
All right, and like you mentioned, yes, I'll be talking about the post-fire restoration at the Bolsa Chica Ecological Reserve today. Thanks. Oh.
All right, we're looking at-
So I'd like to take this time to acknowledge that this study did take place on the ancestral lands of the Achaman and Tonga peoples. So like it was mentioned, in 2020, there was a wildfire that happened at the Bolsa Chica Mesa, and 62 acres had burned down. And although this was an extremely unfortunate event, it did kind of raise a question of we could restore this site. Because before the fire, it was kind of riddled with non-native invasive grasses. And so this came about an opportunity for us to implement restoration techniques and hopefully bring it back to a better state than what it was. And so we worked together with the Bolsa Chica Land Trust to come up with 3 coastal sage shrubs, and 3 native bunch grasses that we wanted to kind of be the focal point for restoring this site. We kind of used the surrounding area to come up with these species. So we have the black sage, California sagebrush, California buckwheat, for our grasses, the purple needlegrass, the beyond grass, and the foothill needlegrass. So those were our main focus for this experiment. And so for our questions, one of the main things we wanted to look at was how the use of herbicide affected the overall success of our restoration efforts. Secondly, we wanted to look at how what combination of perennial species planted as container plants had the best success in growing over time. And then thirdly, looking at how adding native fork seeds, how that increased the native abundance and diversity, and if that had was able to suppress the amount of non-natives that we saw out in the plots. Oops. So for our experimental design, we used a randomized block design, and we had 10 replicate blocks. So this is an example of what one of our blocks looked like with all of our different treatments. And so in blue, we have our plots that were treated with herbicide, yellows, no herbicide, and then for our shrubs, the grasses, they were randomly assigned order in each of the plots. And then all the little dots are the ones that were had the native fork seeds added to them. And then so once our experimental design was all laid out, 2 weeks after the first rain event, herbicide was treated on the herbicide plots. And then after following that, was the only time herbicide was treated on the site. But after that, the volunteers with the Bolsa Chica Land Trust helped with hand weeding events at the site. So for our first question of looking at how herbicide was able to help in our restoration efforts, we wanted to look at the mean number of both native and non-native species. And so in blue, we have our native species, and then in yellow, we have our non-native species. And so one thing we saw that was great to see was for our natives, the increase in 2022, which was really great to see, but we didn't see any difference in herbicide treatment. However, when looking at our non-native plants, we did see a difference in this first year, which tells us that herbicide was really great at suppressing these non-natives. But that trend didn't continue in 2022, which is partially because we didn't treat with herbicide again. So this kind of comes to show that continuing herbicide treatment is something that would kind of help suppress non-natives at this site specifically. So something to consider. And then when looking at our next question of what combination of plants did the best, grew the best, we looked at relative growth rate, and we have our 2 herbicide and non-herbicide treatments. And overall, we saw that our coastal sage-grouse shrubs did a lot better than our native grasses. And one of the main reasons is because of the desert cottontail rabbits and the California ground squirrel. They just loved the native bunch grasses that we presented them. So they kind of demolished the grasses that we planted, unfortunately. But our shrubs fared a little bit better. And so we wanted to look at, of the 3 species that we planted, was there any difference between species? So we looked at all 3 of the species that we planted. And although there is a little bit of like little difference in growth with the Salvia mellifera, there was no significant difference between the 3 different species. And this, to note, is just looking at the 2021 and 2022 data. So I haven't been able to look at this year's wet season, so I'm excited to see how, with all this rain we got, that has an effect on the growth rate. So stay tuned. And then for our final question, looking at how the addition of native forb seeds, if that increases our native and non-natives. So on our this side, we have our native plants, and we have the 80% cover. And then in pink, pinkish red, we have our seeded plots, and blue is our unseeded plots. And so when looking at our native plant graph, one thing that we're excited to see was going from 2021 to 2022, we saw an increase in natives, which is great. The seeds that we introduced grew successfully. That was really great to see. One thing we did notice that was a little interesting was the unseeded plots had a fairly high amount of natives, which is great to see, but we just weren't expecting it. We think a lot of the reason why was the plots were fairly close together, and so there was a chance that seeds could have possibly have gotten into those plots. That's kind of why we're thinking there's such a high amount of natives in the unseeded plots. But when looking at our non-native plants, we didn't see a difference in seeding treatments, but we do see a little bit of a trend where the unseeded plots tend to have more non-natives than natives. And so although it's not a significant trend, it is interesting to see. And again, this is only looking at 2022. So getting able to incorporate this wet year, I'll be interested to see if that trend continues this year. And then when looking at species richness, specifically natives, we did see over the years an overall increase in native species richness, which is really great to see. And again, we were a little surprised to see the high amount of natives in our unseeded plots, which again, we think was mainly due to the proximity of the plots, and there being a chance that the seeds made their way into the unseeded plots. So yeah, just to wrap up, for our herbicide plots, we saw that they did a really good job of suppressing non-natives, but it was kind of tapped at that year mark, so it's something to consider in the future is continual treatment of herbicide. Secondly, we saw at the site that native shrubs tended to do a lot better than our native bunch grasses. And thirdly, the addition of native forb seeds had an overall increase in our natives in this site, which is great to see. And I just want to give a big thanks to everyone that worked on this project. Thank you.
Yeah, we can have it.
Is there a reason why those 6 species were chosen and not others?
Yeah, so we worked with the Bolsa Chica Land Trust, and we chose those primarily because based on the surrounding area, those were the most dominant natives that we saw around the area. So we thought that based on being in similar soil and stuff, they have the best chance. But yeah, there's a possibility that others would do well, but based on just proximity and kind of anecdotal evidence, that those seem to be kind of what had been there in the past.
herbicide were you using?
Ooh, it was diluted glyphosate. And that's what CDFW, they were the ones that, they, treated the area with the herbicide, so yeah, they helped with that.
What was the predominant non-native originally or coming in? Was it on the drones? Or?
It was, yeah, lots of bromus, but one of the main non-natives was the wild radish. That was one that is in the Bolsa Chica area, the big crop, but lots of bromus was also, especially for grasses and mustard as well. But yeah, the wild radish was definitely the most abundant on you. So.
Yeah, one more. Go ahead.
Has that gonna change this total chemistry or composition? What area was?
Yeah, that's not something that we've looked at, but I'd be really interested to see kind of, especially with fire and stuff. And looking at the native seed bank too. But yeah, that's something we haven't looked into, but something that I'm sure would lead to really interesting stuff. Yeah, sadly, I don't know.
Thanks, Adrian. Okay, so our third speaker, Mel Letterman. Graduate botany student at Cal State University Fullerton, working with Joshua Bearlap. Mel is currently involved with several extracurricular botanical exercises, including herbarium assistant at the Niveditha Herbarium on the campus of Fullerton, program technician at Irvine Ranch Conservancy, and native plant specialist at Tree of Life Nursery. Mel is also a member of this chapter, so if you look familiar, that's why. The focus of this presentation will be on documenting the flora of native and exotic, particularly unexamined area where, right in the middle of northern Orange County, despite development all around it, there's only two herbarium records for that area. And the presentation title: Floristic Study of Brea Dam Park, Fullerton, California.
Hi, Ron.
Hi.
Oh, hi.
You want some help?
Yeah.
Okay.
Oh,
Or-
it's start on its own.
No, I have to start it. You need to tell me when to
Oh,
start.
yeah, you can start.
Okay, thanks.
Cool. So I am doing my project on the Brea Dam Park that's located in Fullerton, downtown Fullerton. You just wanna put this
Hold on.
Okay, so Brea Dam Park is located in pretty much downtown Fullerton. It is in between a lot of development of housing. It is a really degraded park. It's approximately 224 acres large, and it was built, so the Brea Dam, it's a recreational space built around the Brea Dam, which was built in 1940 and finished construction in 1942. So it was permitted early 2000s by the state, or Army Corps of Engineers permitted the city of Fullerton to create a recreational space. And it's primarily used as a flood control area, but there is recreational spaces for like biking, walking, running. It's really degraded with a lot of non-natives. And since it's primarily used as a flood control basin, the vegetation is not really considered that much. It's usually managed to mitigate fire risks. So you can see this whole area has been cleared. It was a lot of like grass and nigra. So still a lot of non-natives, but there's a lot of clear cutting in those areas because there's so many homes in that area adjacent. And then this is a photo of the Brea Dam Bilge. That before photo at the very beginning was me standing on top of the dam over there, and there was no water in it, so this is completely flooded. This photo's good. It was from the 1970s. It's definitely not from the 1970s. The cars are like early 2000 cars. So this is definitely a very recent photo. So this is the watershed of the Brea Dam. So it's 22 square miles of the Brea Creek. So it starts right here up in Twin Hills. And then the gray area all around there is that watershed. And then right here at that really intersection right there, purple and blue, that's where the Brea Dam is and where the Brea Dam Park is. And then continues into Coyote Creek, this blue, and then connects with the San Gabriel River and then goes out to the ocean. So that's the full map of everything. And this is from USGS. So they partner with the Army Corps of Engineers to do these water. So I chose this project because there are very little herbarium specimens. This is a screenshot from CCH2, which is the California Consortium of Herbarium. And you can see all of these orange points everywhere are outside of the line of the park. So the line of the park is this triangle. So it's Bassenturi, Harbor, and Brea. Those are the avenues, the streets. And down here are actually two of Bob Allen's from the 80s, his herbarium records. But everything is from like pre-80s on. So it's like 1940s, there's the 60s, nothing really recent. They're all probably geo-referenced. So nothing's probably like even within that area. And I'm focusing mainly on natives and naturalized plants. So there's a lot of cultivated plants that are in there that were just planted in there. But I'm recognizing only the plants that are native and naturalized with the Jepsons, so they're payable and everything. And I'm going to be putting my collections into the Fay and McFadden herbarium, which is Cal State Fullerton's herbarium, and with potential to reach out to other herbariums if they'd like to get some records. And as of last weekend, June 18th, I have 47 collections throughout the park, native and non-native plants. So I made an iNaturalist project along with this. There's a QR code that links you to the page if anyone's interested in looking at it. But this is a screenshot of the page as of I think yesterday or the day before. And before I started this project, there were very little observations being made within the park. And so you can see I'm the top for both of these spots now, but before that I have 107 observations since about May, and then 90 individual species. Before that, was Crystal Watchers and Josh Durr were the kind of two leading people making observations within the park, and not to bag on them, but they're really low observations, and the first observation was made in 2017, so that's not a lot of observations. So in total, there's 237 total observations from all of the public, and that's 30 community scientist observations being made. Those are the individuals that made those observations. And there's 132 species within those 237 observations. So then it started March 2017 was the first observation made by Josh Durr, and then to present. So I made this polygon, and it kind of gives a little bit of extra wiggle room within there, because there are baseball fields, there's a golf course at the very bottom, there's the dam right there, big concrete part of the dam. So I gave myself some little like wiggle room within this project so that if I wanted to go a little bit outside of maybe like the limits of the park. And then this is just a brief taxonomic summary of everything. So total observed by everybody on INAT, there's 48 different families, and then I've observed 42 on INAT, so that's 88% of the families being covered. 111 genera, and then I've gotten 86 of those, so 77%. And then with species, there's 132 different species in the park. I've gotten 90, so 68%. And I've vouchered specimens, so I've collected specimens, 47 of those, making 35. So I still have about another year left of this project. I started pretty late. I started in May of this year, so kind of towards the end of the flowering season, but there's still a lot of stuff there just because phenology has been really off this year. And then some notable finds that I found at the park, just because there's really nothing documented there. There's a lot of individuals of Juglans californica, so the Southern California black walnut. I got to see it with the male flowers right here, the staminate flowers, and then these are the fruits that were like last weekend. So that was really exciting. So that was the female fruit or the female flowers at one point. And then I found one single Asclepias areocarpa in a really sad area. That's it right there about a week ago, and it's pretty clear cut. So I haven't vouchered that because it's really the only one and it's really like sad in that area. I wanted to be able to see it, so I don't know if I will collect it, but I am really excited on that find. And then I kind of do a lot of like going in the gutters and looking around for everything. And I found this Mirabilis labius variation crassifolia, so the California wishbone. I was really excited to see that. It was in full flower. It was in a really random spot. And so I don't think it was planted. I think it may have come in through the drain or something. So really excited about that. And then two more that I'm really excited about. The Actistrong glaber variation vermillatus, so the short-winged deerweed. I haven't really seen it, so it's not very coastal, not abundant around here. The closest I've seen has been in Casper's Park, so I was really excited to see that. I only saw like 1 or 2 individuals in that area. And then there was a lot of the Euphorbia albo maritima, or marigina, albo margin, I can't say it, sorry. But the white margin saying that. So a lot of that Euphorbia was in this like area. It was very close by that Asclepias, but was really excited. It's a very disturbed area, but I was really pleasantly surprised that there was just a really big patch of it. But yeah, these are my acknowledgements. Thank you to people that have helped and other people in INAT that have ID'd my stuff. Yeah.
Yeah, let's have a question too.
Are there any species that you're looking for that are not found in the area? Like any species that are like on the perimeter or in Coyote Hills you're looking for?
Not really. So I originally wanted to do my project in Coyote Hills, which is another area very close by. A lot of it is owned by Chevron, and they're slowly giving bits and pieces to the city of Fullerton. They didn't want me to they didn't want to give me a permit for that because there's California gnatcatcher habitat there. So they're really hesitant on giving me access to that. I'm kind of guerrilla, just doing that in here. So just because it's a really disturbed park, nobody's really like paying attention to like all the weeds that are everywhere, all the non-natives that are there. So no, there's not really anything. I'm just kind of trying to see what I can get in there and give it a little bit more of a recognition of the plants there. Yeah.
Where are you maintaining the checklist, and is it maintained on CCH2 or is it?
No, so I just have an Excel on my own computer, so that could be something I look into and making a checklist on CCH2.
We can do that.
We can help you.
Sounds good. Yeah, I'll be able to do that. Yeah.
You mentioned about Brea Dam Park being a potential restoration area. Is that something that you talked to city or anyone about?
No, that's just in my own brain, just because it has a lot of potential. There are like a lot of chunks that look like they've been planted over time, but in that one photo at the very beginning where it's just very clear cut, could definitely be like potential instead of just having crews come in like every year or so and just clear cut. I think it would be really nice as like a restoration potential.
Thank you. That was good, Mel. Very good. The next speaker, Niveditha Ramadoss, PhD candidate in evolutionary biology in the joint doctoral program of San Diego State University and University of California, Riverside, working in the Uvia Flora's Renteria lab. She did her undergraduate degree in biotech in her hometown in India. While there, she studied the antimicrobial activity of native plant extracts. That sounds fun. She did her master's at California State University, Northridge in molecular biology, where she studied stress tolerance of plants. Another interesting bit there. Currently, she studies the evolutionary ecology of our native cholla cactus. This presentation will inform about the ecological consequences of having sexes separated in some species of native, in this case, Cylindropuntia, and she's going to highlight wolfii which straddles the border and is a rare plant. Presentation title, Consequences of Being a Unisexual.
Can you all hear me? Yes. First of all, thank you all for being here for my talk in the middle of the week, evening. Thank you so much. Good evening, everybody. How are you all doing?
Great.
So I'm from San Diego State University. I'm doing my PhD with Dr. Flora, and I'm here to present a small part of my PhD thesis, which is consequences of being a unisexual. So sexual reproduction, as we all know, is a universal feature of eukaryotic organisms, right? And for reproduction, we need male and female functions. And how these functions are distributed across organisms of a species is what we call a sexual system. In some cases, you can have those functions on separate individuals, like on lion and lioness. And in some cases, you can have male and female functions on the same organism, like in hibiscus. And when we talk about flowering plants, sexual systems are so diverse. The most common sexual system you would observe is this hermaphroditic sexual system, where male and female functions are present within the same individual, like in this hibiscus. An interesting development in the evolution of sexual system in plants is the development of dioici, or separate sexes. So here you have male individuals separate from that of the female individuals. Like in this kiwi plant here, male flowers will be on a separate plant and female flowers will be on a separate plant. And there are several consequences to dioici, and I'll highlight one of them, which is important for species that have biotic pollination or depend on animal pollinators. What happens here is that you have a female plant and a male plant, right? There could be intrasexual competition between males. Males would be fighting with each other because their success depends on how much pollen they disperse, right? How many females they can get to fertilize. So males will start fighting for competition, and what happens as a result is these male plants could become more attractive to the pollinators because they want to spread their pollen, right? So they might become bigger, brighter, or produce more scent, become more, produce more sweet nectar, something like that to attract the pollinators more. And this is what we call this. What will this lead to is differences between sexes. So you can see how male now looks a little bit different from female by looking bigger. So these differences is called sexual dimorphism. And these differences are also commonly observed in animals like lions. You would be able to look at a lion and say whether it's a male or a female, right? And how do you say that? Using those sexually dimorphic traits. So same thing with plants also. And what happens as a result of all this sexual dimorphism is that the bees will get, or bees or pollinators will get attracted to the males more. So this will create a bias in pollinator visit between the sexes. And this makes the populations of size of the species becoming responsive to pollinator density, as a result affecting their genetic diversity and affecting reproduction. And a type of sexual dimorphism called sexual dichromatism is what we call the distinct differences in color between both the sexes, like you can see in these figures here. And this is common in birds, lizards, and fishes, however, not being reported in plants so far. So one of my goals in my thesis is to explore sexual dimorphism as well as dichromatism in Cylindropuntia wolfii, commonly known as wolf's cholla, which is a rare, native, endemic species of the Sonoran Desert in the USA. And like Dan mentioned previously, it borders USA and Mexico. All the red dots you see here are observations of wolfii. My field is located at this red balloon here called the Mountain Springs area, where the species is predominant, like you can see from the figures in the video here. Two reasons why we chose wolfii is because the sexual system is functionally dioecious. So the plants will either function as males or function as females. And how can you tell the difference between males and females is by the presence of pollen. So male plants have the flowers with pollen, and female plants have flowers that do not have any pollen. That's how you can tell the difference. And second reason, the beautiful diverse flower colors that you can see in this species, which is all located within the same location. So we have 6 different flower color morphs ranging from green, then you have something between green and yellow, yellow, orange, red, and so on. So 6 different color morphs, which will help us to study dichromatism. But wolfii has a reduced seed production. The females rarely set any fruits with mature seeds. So mature fruits are look like these, they're round and plumpy, healthy with seeds. Aborted fruits are like this, and this is what we mostly see in the field, which suggests that the sexual reproduction of this species may not be very efficient. And we just saw how sexual dimorphism could be influencing reproduction. So that's why we ask the questions, is there sexual dimorphism in wolfii? If so, are the flower colors associated with a particular sex, which is dichromatism? And are there other sexually dimorphic traits? So first question, are color and sex related in this species? We did a transect study, a 600-meter transect study with 80 plants. Did a chi-square statistical test to see for significance. And we did see a significant relationship between sex and color, with males usually being green and yellow towards the brighter side, and females usually being orange and red towards the darker side, suggesting there is sexual dichromatism in this species. However, there's no strict cutoff in terms of dichromatism, because sometimes you can see males being red and females being green. And how we can explain this is that brighter colors are good on one side because they attract more pollinators. And this trait is especially advantageous for males because, like we discussed, males' reproductive success depends on how much pollen they disperse, right? Whereas brighter colors, on the other hand, can also be bad because they'll not only attract pollinators, but also insectivores and florivores and herbivores, right? And this disadvantage is especially bad for females because they need to produce the fruit. So they can't afford to lose the flowers to florivores, right? So this is how we think, why we think females usually stick to the darker shades, while males usually stick to the brighter shades. And then we looked at other dimorphic traits, like quantitative traits. We looked at the diameter of the flower, measured the diameter, tepal length, which is basically the petal length, and then tepal width, and followed by statistical test to check if they're significantly different between sexes. And followed by, here are some of the results. On the x-axis, you see both the sexes, female and male. On the y-axis, the different quantitative measures. As you can see from these graphs, males have a significantly higher value compared to the females, which suggests that the male flowers are bigger than females. And this brings us to the hypothesis that there could be a bias in pollinator visitation, with males being more attractive to pollinators than the females. And bigger and brighter male flowers are common in other dioecious species also, because larger flower size and brighter colors are usually selected by pollinator-mediated selection. And again, like I mentioned, this is advantageous for males because their reproductive success is limited by the pollinators. And other traits that are only visible by our bee pollinators, traits like UV and green fluorescence. We measured these traits using Nikon stereo microscope. And here's a picture of how a yellow flower looks from human side view versus a bee side view. So when we measured UV, we didn't see a significant difference in terms of for both the sexes. But then for fluorescence, we saw a difference. So you can see this is a female plant and female anthers, male flower with male anthers. You can see that the females are more brighter, right? More shinier compared to males. Yes. So this GFP, which stands for green fluorescence, was definitely brighter and significantly brighter than the males in terms of their emission intensity, which suggests that there is fluorescent sexual dichromatism in this wolfii species. And what this tells us is that these signals have known to be involved in plant pollinator signaling. We already know honeybees are attracted to blue fluorescence and that bees can see green fluorescence. So we hypothesize this green fluorescence in females could be attractive to the bee pollinators that possess this green light attractivity or sensitivity. And this could be one way how females are compensating for their smaller flower size and darker flower colors. And interestingly, fluorescent sexual dichromatism has only been observed in birds. The first and only study was by Brea et al. 2012 in these blue-winged parrotlets where males were fluorescing instead of the females. And so we hope this finding could be a foundational study for sexual dichromatism as well as fluorescent sexual dichromatism in plants. With that, I would like to acknowledge my institutions, my PI, Dr. Flora, collaborators, committee members, funding sources, my lab members, and all of you for being here. Thank you so much.
Maybe great talk. Within female flowers, have you looked at if there's a relationship between flower color and seed set?
Flower color and seed set. We did measure the seed set of different female flowers, but I don't think we saw a significant difference. That was my chapter 1. I don't think we saw a significant difference. They were mostly aborted for one, and yeah.
Can I ask another question?
Yeah. Sure, yeah.
Have you thought about pollinator fidelity and how so a bee needs to visit male flowers and female flowers in order to affect pollination? If they're focused on collecting pollen, they're going to be going from male flower to male flower to male flower, and won't be visiting the female flowers unless they're then switching towards her nectar. Have you thought about how that might play a role in the low seed set we're seeing in the cactus?
Right, we think there could be an influence for the nectar, like you mentioned. Maybe the females are trying to compensate for their absence of pollen by having a more sweeter nectar, or maybe even higher floral scent, or maybe this fluorescence of the anthers, or also the darker flower colors make the anthers, the yellow anthers, stand out more. So maybe compensating through all these techniques, but we haven't really measured them. But that could be a future work in the species to see if that's where they're compensating. Yes. Okay. Yes.
I guess to follow up on that, do you know what mechanism caused wolfii to become dioecious? Because I know like most cactuses that I see here are mostly like monoecious. And it seems like if a trait is evolved to be like beneficial for one sex, but it compromises like the success of the species, then it doesn't really make sense for the species to go from monoecious to dioecious.
Yeah, that's a great question that I've been thinking about, like how did they evolve to be dioecious? We have so many hermaphroditic cacti, and there's also some gynodioecious species that are putative close relatives to wolfii. So I assume that could be a transitional pathway from going from hermaphroditic and gynodioecious, where you have females and bisexuals, and then female to male dioecious. And that has been through other species phylogenetic studies, they've shown that this could be a transitional pathway. Gynodioecious could be a transitional pathway. So, but if we get our genetic data soon, then we'll be able to build a phylogenetic tree, maybe trace the sexual system evolution. Yeah, that would be a great future work.
Very quickly, so just to clarify, the aborted fruits, are those fertilized or are they unfertilized?
They could be unfertilized, or maybe if they were fertilized, maybe if they had just one pollen being dropped and fertilizing, the plant would just abort it because it doesn't want to spend all the energy for just one seed. That could be fertilized or unfertilized, yeah. Or lowly fertilized, like not enough fertilized for it to develop, yeah. Yeah.
Provoking. So tonight, fifth speaker, Daniel Donovan, received his master's just recently from Cal State Long Beach Ecology Department. That was February. Working with Amanda Fisher from her lab. He was the 2021 Charlie O'Neill grant recipient from this chapter, by the way. He's now serving as the field biologist, or botanist rather, for the San Diego Natural History Museum. So he's running into John Redmond a lot down there, so that's good. Daniel made it his quest to find and kind of document flora on a very difficult part of Orange County, Lad Canyon. 79 field trips, rare plants, almost 700 new voucher specimens, but he'll tell you about all that. Presentation title: The Vascular Plant Flora of Lad Canyon, Santa Ana Mountains. Dan. I get it on there eventually. There we go.
Sorry, I didn't know your.
Sounds good. All right. Pointer. I think it's the yellow button. The yellow button. Oh, it's up there. There you go. Right. Yeah. Okay. And that's in. Well, thank you, Dan. There we go. Hold on. Hold on.
We're fighting each other. No, go ahead, try it again.
So I'm gonna spend my time thanking everybody on this list. And then when I'm done, I'm gonna thank the people that didn't make the list, like Wayne. But honestly, I just want to say I really love the chapter and I really appreciate the support. I know that you guys already are well aware of the California Floristic Province and probably the Santa Ana Mountains as well. I'll just tell you that Lad Canyon, the focus of my research, is just north of Silverado Canyon. And here's a closer look at the canyon. The main access, is along Main Divide Road here. There's also some access on Silverado Trail, a lot of switchbacks, and then Ron B helped me get some access from private land at the lower canyon, which was a huge help. Thanks, Ron. Also, I need to tell you that in the early 2000s, Southern California Edison installed some power lines on the ridge here below Pleasant's Peak, and there's a service trail that was added for those power lines that was useful for the research. Not a great trail, but the mountain bikers apparently have used it and called it the special trail. The main habitat in the canyon, chaparral and coastal sage scrub. There's also some grassland. How's the sound been? Okay. Yeah. So the grassland is non-native grasses, but you do get a lot of native herbs in there. The canyon bottoms have a lot of great trees, big Doug fir, canyon oak, big leaf maple, a lot of things like that. And it's beautiful in there. You can see that it's really relatively undisturbed part of Orange County, including a vulnerable turtle here, the Western Pond turtle. So what is a floristic survey? Basically, it's an annotated checklist of all plants that are in an area. And it's based on voucher specimens like you see on the right here. This is a specimen dried and pressed and preserved on a herbarium sheet. And they can be preserved for decades and even centuries this way. And they are the main evidence we have for what plants exist in an area. They're essential for botanical research. And all the data that we collect with the specimen is reported and is made public on the California Consortium of Herbaria Database, CCH2. And here are some of the previous surveys that have been done in the Santa Ana Mountains, including importantly the preliminary survey of the entire Santa Ana Mountains by Elastic and Thorn. So why did I choose to study Ladd Canyon? First of all, because it is, relatively speaking, a botanical black hole. Not a lot of botanists have made it into the canyon. And then just in general, floristic work is, very important for understanding the distributions of species and in fact finding new species. And it's an important baseline evidence of what we have as things change. And it's very important for land management, especially the finding the presence of rare species. And then there were specific historical and physical characteristics of the canyon that also made it interesting that I might have time to touch on. So there were previously 282 voucher collections made, mostly along Main Divide, and you see that some recent ones along Smash Mouth Trail. I think those were all by Cal State Long Beach students. And I made it a goal to visit every 1 kilometer square in this grid of the area. And you can see I basically did pretty well. This is most of my trips, not all of my trips were recorded. There's actually 8 field trips, whatever. So this is a 3D view, give you some sense of the terrain that I was going with. And I did make 695 new collections, pretty well spread throughout the canyon. I also created an iNaturalist project. About 3/4 of these observations were I made during the project. And so here are some of the results. The checklist includes 324 total taxa, which I think is pretty good for a relatively small area for a super like this. And these top 5 families that you see here are not a surprise. These are big families in the California forested province. So one interesting thing about the area is the Navajo pine stands on the higher ridges. They are seemingly completely associated with serpentine soil. The Pleasants Peak area at the north of the canyon is the only area in the Santa Ana Mountains that has a serpentine soil, which is very harsh environment for plants. It's low in important nutrients and high in magnesium and metals that are harmful. You can see, so I visited these different stands, and you can see how in the middle of this serpentine outcrop, the trees are very small and a lot of yellow needles, whereas farther away you get these healthy tall trees with lots of cones. Another interesting thing about the history of the canyon is a relative lack of fire. The normal fire interval in Chaparral is like 30 to 70 years, and some of these, a lot of the canyon has never had a recorded burn, and some of it, most of it hasn't burned at all in more than 70 years. I can't get into the research of all what this would mean, but the basic upshot is that you're likely to have fewer annual plants when you have these long fire intervals. And in fact, this is what I found when I compared my checklist with checklists for, that's kind of slow, for the checklist for the Santa Ana Mountains as a whole and for the San Mateo Canyon in south of Ortega Highway in the southern part of the mountains. This is with the caveat that I was collecting for 2 years. They weren't the wettest years at all, and so that could affect, but this is somewhat suggestive of what was going on. Another interesting thing that was talked about in a previous paper on the ecology of the Navajo pines was that there's a different microclimate in the canyon, and that moist marine air was forced into the canyon, making it wetter and helping the pines. I looked at data from Southern California Edison weather stations, spread throughout the mountains. Some of them which just only came online during my project. And basically what I found was that this hypothesis wasn't true. It's not wetter than Upper Canyon. There's a trend of wetter at lower elevation, drier high elevation, and my area was below the trend line or near it. Third thing about the canyon is not a lot of people make it in there, and so there's less human disturbance, despite what we see. This is up near the main divide, of course. And that's likely to lead to fewer non-natives and invasives. And looking at, again, in comparison to those same areas before, I did indeed see fewer non-natives. So I like to end with the cool plants, which is really what it's all about. Up along the ridges, there's Lightening cardiophylla, which has its largest populations in the Santa Ana Mountains. It also goes down into Baja, but there were great populations in the area. It's a wonderful smelling plant. I highly recommend it. When I started, it was covered with, when full bloom, it was covered with these mason trees on the right. You see. California's weedy intermedius is an Orange County favorite. It's intermediate between California's Plumery to the north and weedy in San Diego County. And it did pop up in the area. A third rare plant is Monardella hyperluca intermedia, and very similarly, it's intermediate between the subspecies to the north and a subspecies mainly found in San Diego County. This one I found 7 different populations. It showed up in chaparral understory, in porcupine understory, in riparian area. It's another one in the mint family, a neat little plant. And this one is actually, this is like breaking news here. So I went back to this specimen last weekend because I've been learning some and working on the job, and I wanted to revisit this one. And basically what we're seeing here is, so this here is Allium maticiden, and this is Allium Mardinii from the Jepson manual. These specimens, when they're dry, in the field, you might not know what you're looking for. Back in the lab, it might be dry and a little bit hard to figure out at first. It really helps if you know exactly what you're looking for and you're familiar with these plants, but I'm pretty certain that this is a rare Allium Mardinii. The humble lily is a watchless plant, and it was I figured there's more than 1,200 plants in the canyon and all parts of the canyon. It's very charismatic plant. It's fun to see, and unfortunately, I think part of the reason you don't see it more in other parts of the canyon is because of they're so attracted to humans. Basically, don't pick flowers, people. Another big charismatic plant here, the Theo poppy. For some reason, I was mostly just finding it in the West Fork. This guy was everywhere. I don't know why to watch this plant. It's everywhere. It's like a weed. And this is I only found this in a couple places. It's just a very, very small number of it in the riparian areas. It's a beautiful flower. And lastly, I've got this interesting hybrid. I just saw a couple of these plants. It seems to be a hybrid between Cynopus papillosus, which you can see on the right, and the Cynopus eleganthus, which is much more common in the Santa Ana Mountains. Some of the characteristics, let's see, you can see here that the glands, normally in Eleganthus, it's just on the margin of the leaf, but it starts to curve here on the axis side of the leaf too, similar to how you see with the Cynopus papillosus. And that's all I have to do. More pretty flower pictures. Thank you.
Quick one out. He answered them all and.
No question, but I just acknowledge how difficult that work is, but. Daniel did, the number of visits and the number of collections and the terrain, Daniel, that's amazing. Nice job.
Well, thanks, Brian. Well, thanks a lot for helping me get the access, because otherwise I'm going up with the Silverado Trail and then down the side and again, over and over again, so thank you.
No. A minute or two.
Okay.